My name is Rodrigo Morales from the University of Texas Health Science Center at Houston. And the title of the project that we are planning to pursue is the development of a high throughput system for screening anti-prion molecules. So why we are studying this? So as you know, prion diseases affect humans and several animal species, but prion diseases is not one disease. As you know, in humans, for example, we have different types of Creutzfeldt-Jakob disease. We have Kuru, we have fatal familial insomnia. So all these diseases are thought to be caused by different strains of infectious prions. So what are these strains of prions? So we believe that there are different conformations of the infectious agent, different shapes of the infectious agent. So if we think on these different strains as different conformations of prions, it is easy to think that a certain drug attacking one conformation will not be doing it in the same way for a second strain, having a different shape. So that's why we believe that, for example, some previously identified anti-prion molecules has been again identified in rodents have not been active in for human prion diseases. So what we are trying to do in this project basically is to develop a system that will allow us to screen libraries of small molecules with the hope of identifying strain-specific anti-prion molecules. So for that, we are taking advantage of the PMCA technology, the protein misfolding cyclic amplification technique, that is able to replicate prions in a very fast manner. And fortunately, this technique also is able to maintain the conformation of the infectious prions that it is replicating. So in that sense, we can test whether a specific compound is able to inhibit the propagation of a specific prion strain. So in this project, what we are planning to do is to work in two different aims. In the first one is to standardize this high throughput PMCA platform that we call for two different animal species. We are planning to do it for human prion strains, but also for deer prions. So of course, the relevant thing here is to do it for human prions, but it's critical also to do it for deer for two reasons. One is because of the problem that chronic wasting disease prions are making in the North America, especially in the United States, but also is because we want to test whether molecules identified as active for human prions are also able to be active for deer, or they are exclusive for humans. And the other way around, we want to see whether some molecules active against deer prions are able to go against the human prions or not. So we will develop this in two different formats. Hopefully we will go for a 384 well-plate format that will allow us to test a higher quantity of molecules. And once this is standardized, what we are planning to do is to screen these large libraries of molecules in this high throughput PMCA technique, with the hope again of identifying strain-specific anti-prion compounds. So what we have done so far, we have standardized the high throughput PMCA technology for 6 different rodent prion strains, and we have identified some strain-specific anti-prion molecules. So we know that this principle works using specific and well-established prion strains generated in the laboratory. So considering that, we want to jump now to the next step, testing the same principles for more relevant human prions and deer prions. So with that, I would like to conclude. I would like to thank the different awards that are making possible this research. And I really hope to bring good data and good results for the next year meeting. Thank you very much.